So welcome back guys, welcome to another lecture series of translation in prokaryotes. And in the last lecture we have already studied about the initiation of translation, and today we'll go with the elongation part. Okay, we'll go with the elongation of the translation process. Translation also you can say about the protein synthesis. Okay. So, starting with the elongation part, we need to understand what are the different steps and how it begins. Okay. So, when you see the 70S complex, as we have already studied, that there are two different units formed and they together form the 70S complex or the 70S unit. In the large subunit we have the 50S, and in the small subunit we have the 30S. So these two together combines and form the 70S complex of the ribosomal rRNA. Okay. So here you can see three different sites. If you have already seen my last lecture, you can understand this, because I have already mentioned that the E site, P site and A site. So if you haven't seen that video, just go back to that video and watch it and then come here, so that it will be much more better for you to understand. So now you see here the ribosomes contain three different subunits, E, P and A site. Okay, three different sites, not subunit. Subunit is 50S and 30S. So in this three site, the first tRNA, this is a tRNA, this is a tRNA, and here you will see the tRNA that comes first will be attached with the amino acid here, that is formilated, that's a modified amino acid, and they will come and bind with the start codon here. Okay, and here in the A site and the E site they will remain free. So now before starting the process, you need to know that the steps, so that you can understand it much more better, in a much more better way. So in the first step, what happens? This tRNA that comes in the A site, the A site will bring one amino acid with it, and that will be particular to that specific region of the mRNA in the A site. Okay. Now try to understand this step. Try to understand what it has written here. The tRNA that will come here, suppose here one tRNA will come at the beginning of the step. Okay, here they will bring one amino acid, that means they will be charged, and this tRNA that brings the amino acid will be binding to these sequences out here. That means there will be another codon here, and this codon, anticodon, will be interacted here. There will be interaction between the codon and anticodon side, and accordingly according to this codon they will bring the amino acid here. Okay, and they can now start the elongation process. Okay. So now you see in the first step, each of these tRNA that I have told just now, that will bring one amino acid particular to the specific region of the mRNA in the A site. Then in the next step, what happens? The attachment of amino acid with each other via peptide bonds. So now what happens? In the next step, there will be attachment between these two amino acids. Here one amino acid will be there, and here another amino acid will be there. That will be already from the beginning. Okay, they will get attached here, or they will form a peptide bond here with the help of an enzyme known as peptidyl transferase. And this peptidyl transferase activity will be done with the help of two different they have different groups there. Okay, they have amine group and also they have carbonyl group here, and this amine group of the first amino acid will attack to the carbonyl group of the carbon, and they will form transpeptidation reaction. They will form transpeptidation reaction through which they will form peptide bond. Okay, so this is the second step. Now coming to the final step, where the ribosome will slide along one codon. The ribosome will slide along one codon from 5 prime to 3 prime. Suppose this is the mRNA out here, 5 prime, 3 prime, and this will be the ribosome. Okay, and from the start side they will move in the 3 prime direction, and what happens, this ribosome will move forward by sliding along one codon unit, or the 3 nucleotide sequences. Okay, 3 nucleotide sequences. So one codon means 3 nucleotides, so they will move one codon unit from 5 prime to 3 prime. So this will be the final step of elongation, and after this they will keep on repeating this cycle until they reach the stop codon. Okay, until they reach the stop codon, and after that they will when they will reach the stop codon, they will start with the termination process. So now let us see this process, let us understand this process much more elaborately. See, in this ribosomal unit, in the large subunit, you will find translation factor binding site. If we begin with the elongation process, at first what happens, the tRNA that needs to come and bind to the A site have to come, but they cannot come directly. They need some mechanism, because that doesn't have much energy to move and go and bind with the A site. So this tRNA needs to bind with some elongation factors here, and nearly 3 elongation factors are there in translation in prokaryotes. First one is EFTU, which is a GTPase protein, and the second one is the EFG, elongation factor G, or the this one is also a GTPase protein, and the final one has some different activity which I will be telling you a bit later. And this final one is EFTS, or the elongation factor TS. So now at the beginning what happens, this tRNA will come and bind to the A site with the help of EFTU, because this EFTU has GTPase activity, so they can come here and bind. And in the next step what happens, after binding this tRNA along with this amino acid, the charged tRNA, that means charged tRNA means the tRNA that has amino acid bound to it. Okay, so this tRNA will, suppose this is the tRNA, so this tRNA comes and binds in the A site, and after binding in the A site with the help of EFTU elongation factor, because this elongation factor has a GTPase activity, they will provide energy by hydrolyzing this GTP into GDP plus inorganic phosphate. So they will hydrolyze its GTP and provide energy to bind this charged tRNA to the A site, and they will now dissociate. This EFTU becomes dissociate after its activity is done. So now what happens, again another tRNA comes in the next step, and again this EFTU will come, But this EFTU cannot do the process repeatedly without having this GTP activity, because this EFTU has already hydrolyzed and they have converted into GDP plus inorganic phosphate. So they need to be again converted or to be recycled into GTP. This activity is done with the help of EFTS factor. The last factor which I have already mentioned that is this EFTS has the activity of recycling the GDP into GTP. They will convert this GDP into GTP and they will provide the EFTU with the GTP activity again, and they can again come in the next cycle and they can provide the tRNA the energy to go and bind with the A site. Okay. So now you see this EFTU has released, and now another mechanism starts, and this mechanism is addition of this peptide bond formation. This peptide bond formation between the previous tRNA and the new tRNA. Okay. The previous tRNA and the new tRNA. It was bound in with the tRNA. That means the tRNA was charged. But now see, after the peptide bond formation with the help of peptidyl transferase, this formulated methionine or the amino acid, previous amino acid, is now bound with this new amino acid. Okay. So this energy is provided with the help of transpeptidation. Peptidation. Okay. I'm just writing it roughly, but try to understand this. So now what happens? Actually, here I have written this EFG factor which comes and bind, but this EFG factor comes later. Okay, not now. First, the peptidyl transferase will add this peptide bond between two amino acids, and in the next step only this EFG will come, the another factor that will bind with the 50S subunit of the ribosome. They comes and this EFG or the elongation factor, they comes and bind with the binding site of the 50S subunit. They cannot come and bind randomly. They needs to bind to that binding site of this elongation factor. So what happens? The purpose of this EFG also is to provide energy. So they have a GTPase activity and they will hydrolyze and they will provide GDP plus inorganic phosphate, and during that process they will provide energy to the ribosome to move one codon forward. So now you see here the actually in the start site there was a tRNA before, the charged tRNA, but after the binding of this formulated methionine or the amino acid with this new amino acid, it becomes uncharged because the bond between this tRNA and the amino acid is broken. So now in the next step what happens, see, as the ribosome is moving from 5' to 3', one codon at a time, so P side will move to the E side and A side will move to the P side. So now you see the tRNA that was present here, the uncharged tRNA moved here. This is the uncharged tRNA, and the charged tRNA along with that peptide bond formation, they will move to the P side from A side. And you have also seen this, that the mRNA is moving closer. That means the stop codon is moving in the opposite direction as the ribosome is moving in the forward direction. So the stop codon that was before in the near to this end has come a bit closer to the A side. So in next step, this A side is free. So why the A side is free? Because now a tRNA has to be added here, and this tRNA should be charged with an amino acid. And you know that in the cell there are different tRNA present. And this tRNA comes and bind with the specific sequence in the codon. They need to find some specific sequence, and depending on the sequence of the codon, they will bring different types of amino acid. Okay. So this is how they comes again, and they will bind here with the A side, and they need to bind with the factor here so that they will get the energy. So they will bind here and they will provide the amino acid, and in the next step what happens, the peptide bond formation will be there, and this peptide bond formation will be with the last amino acid and the new one. So in the next step what happens, again this elongation factor dissociates and a new elongation factor will come, which will provide the energy to the ribosome to move forward. So this way the cycle repeats, and finally they will meet this stop codon here. The ribosome will meet the stop codon here, and after reaching the stop codon, the ribosome will now send a signal to stop the process. So this way the termination starts. With this I end my video here. I hope that you understood it very nicely. Next video will come with the termination process. So until then, goodbye, and do subscribe to my channel and also share with your friends. Thank you.